right, I guess we should start then.
Yeah, let's go.
All right. Hello, everybody. Welcome to the second part of Neurotech's workshop, EEG feature extraction. And so, if you haven't checked us out yet, you could check out our website. You could follow us on Facebook or join our Discord channel. So a little bit about our club, in case this is your first time attending our events. We are affiliated with Neurotech X. We started in January 2020. Our faculty advisor is Professor Vikash Gilja of the ECE department under the TNEL research group. We have connections with a neurotech headset company called Wearable Sensing. Our club also participates in annual student NTX competitions, and our mission as a club is to bring together students interested in neurotech and provide a way to get involved in the technology. All right, so our plans for today are to do refresher about the competition, data set, and what we did last time. We will also be going over some feature extraction concepts, and then we'll be doing a notebook walkthrough, and then the remaining of the time will be office hours until 8 p.m. So before we actually begin the workshop, we would like you guys to answer to us some of these questions. So the first poll question is, have you ever heard of power bins or frequency bands before?
I think they can also say the second one.
Oh yeah, and then the second question will be, have you heard about the phase amplitude coupling before? So go ahead and answer that one once you're done with the first poll. Okay, so looks like that's everybody in the polls. That's everybody in the polls. So if you're not very familiar with the concept of phase amplitude coupling before, do not worry about it. We don't expect that many people to know what it is, but we will be going over what phase amplitude coupling is in this workshop presentation.
Yeah, don't worry if you haven't heard about power bands or frequency bands either. We'll also go through that. Cool. Looks like we'll have a lot of learning to do.
Right. And as a refresher on our NeuroData Kaggle competition, this workshop right now is the second in this series of 4 workshops, so there will be 2 more related to the Kaggle competition that we and DS3 are hosting together. Refresh on the details, there will be up to $100 gift card prizes for the top competitors in this competition. It will last through week 3 of winter 2021, so week 3 of next quarter, and you can really start anytime that you want, as long as, well, you do it before week 3 of next quarter. And also, the end goal of the workshop is to get a Jupyter notebook that models the data and to help you predict whether someone is thinking to move the right hand versus the left hand. And how to join, we have several links to the Kaggle competition, to GitHub notebooks, and also to our Discord for this competition, to the official Discord for this competition. And those are the links posted into the chat. Are there any questions? All right. Move on.
If nobody has any question, I'll continue. So this is where we left off from last time. It's just a little bit recap. So this is how the experiment is set up. We think of the motor imagery task as thinking of either left or the right hand. It's prompt with arrow on the screen. we also defined how the electrodes are placed, where the electrodes are placed on the head, and we can see it's placed on the temporal sulcus, and we can also see the letters under the electrode, and also where the EOG electrodes are placed. We also learn about the timing scheme within each trial of the measurement. We start with a beep, and then that is followed by a fixation cross presented. Then followed by a cue. And we measure the motor imagery during 1 second after the cue is presented, and that is followed by roughly 1/2 seconds of break. Next slide. Now that after we were familiarized with how the experiment was set up, we also get ourselves familiar with the data set itself. In the Jupyter notebook, we visualize the EEG data in the time domain. We then section the raw EEG data into individual epochs. And then we learn the techniques of changing the EEG data from time to the frequency domain and vice versa. Then we learn about a really versatile tool in analyzing the EEG data called power spectral density, or PSD for short. And using this tool on the data, we found on average, we can see a decrease of mu power in the contralateral side during the motor imagery task. Last but not least, we elaborate on this technique and on a more detailed method that not only has the power of different frequency, but how it changes over time. And that leads us to today's material.
Yes, so today we'll be talking about feature extraction, specifically in the frequency and time series domains. We'll look more closely at the PSDs to see what we can gather from that, and then also see how we can quantify it with bin power analysis, power bin analysis, band ratios, channel differences, time series features such as Hjorth parameters, and instantaneous measures such as amplitude, phase, and frequency. And we can see how we can get that with a NeuroDSP. And next time, we'll also introduce a little bit more features and also how you can use these features in a model to predict whether someone is thinking to move their left or their right hand. Oops. All right, so PSDs are very visually helpful, but they're very hard to quantify if we want to use them as features. So here, last time we saw that we can see how in this alpha range, the C3 and C4 electrodes sort of swap their, like whichever is higher. This is because of the event-related desynchronization, ERD, that is a neural quality that happens when you think about moving your right versus your left hand. So whenever motor imagery happens, that part of the brain sort of desynchronizes, and it lowers the alpha amplitude in those channels. So if you remember that C3 is the left-hand side electrode, just imagine that it's here on your left hemisphere. If you think about moving your right hand, then this ERD will sort of reduce the power in the C3 electrodes, which is what you see here. And then similarly, when you think about moving your left hand, the power on the right-hand side hemisphere of your brain, which is covered by the C4 electrode, will also see a reduced power in the alpha band. And these are, again, mu waves, as we talked about last time. Okay, so how do we quantify this? Like, how do we make sure that the data that we have to represent this kind of information is helpful for our modeling. So one way to do this is with power bins. And this is sort of, you can think of it as the integral of the PSD over a bin range. So in the past, we saw that this is the alpha range here, right? From 8 to 12 hertz. If we were to take the integral of that signal of this PSD in this range, we get a sum of all of these values basically, and we can then get a single value for that information. And so this is easily done with the PyEG bin power method, where if you give it the frequency bands of your interest, it will filter it down to that and give you the power and also the power ratio for those frequency bands. This is what we see down here. So I've broken this down by channel, but you can also like analyze it so that you can see channel side by side in these graphs. But this one here is the C3 channel. And you can see how across different classes in the alpha band, we have a similar thing that we saw before where the left case sees a slightly higher power ratio. So if you think about moving your left hand, then the C3 electrode will be higher because it'll sort of reduce the in the C4. So you can see how in the C4, it's slightly less. And then the opposite happens when you think about moving your right hand, that the C3 electrode, because it's on the opposite, the contralateral side, it will be reduced in alpha power. So this is a nice way you can potentially use to sort of quantify how we can interpret the PSD. Are there any questions? Oh, yeah. So 0 is left and 1 is right. Yeah, as Simon said, a mu wave, yeah, sorry I didn't clarify that earlier, is specifically the alpha power at the motor cortex region. This is specific 8 to 12 hertz. Yeah, for the mu wave, yes. Or it can sometimes, like, people's definitions of alpha sort of change. So it could be like 7 to 12, as in this case, on the slides. Oh, yes. The difference between power and power ratio, that's a very important difference. Power is the actual like integral. So you basically sum up the actual power for each of the time points, or not time points, but each of the like PSD pieces. Power ratio is if you were to do that for each of the power bins and then divide by the total power. So in this case, we see power ratio is actually, they're all lower than 1 because if you divide by the total thing, you'll get a fraction of 1. But if you were to add all these up, you'll get 1 again. Like if you add all the blue ones together and then do the same with the orange ones, you'll get 1. Yes, it has a normalizing effect. And you can sort of think about how that might help or hurt you, because in the case that it helps, it could like sort of reduce some noise in terms of how the amplitude of the EEG signal actually changes over time, but also you lose that information, right? So you could try exploring using either one of them. Cool. These are great questions. Awesome. All right, so we'll move on to the next thing. This is band ratios, which is very similar to using power bins. So we'll build off of this and we'll look at the ratio between the different powers. So if you think about over here, if you were to look at like say theta beta power ratio, like if you were to divide this by this, you can potentially see how increasing theta and decreasing beta could create a different ratio, but then different ratios can also mean different things. So in literature, we see that theta beta has been discussed a lot in the ADHD realm, and also theta theta gamma has been discussed in learning quite a bit, and there's also some other different mental activities that have been discussed with different band ratios. So there's a lot of literature on the different things, but there is also some debate as to how this can actually be interpreted. For example, in the ADHD patients, when they say that ADHD patients have higher theta to beta ratios in their EEG signals, this could also be interpreted as a slowed alpha. If you think about how alpha on the previous slide is kind of basically adjacent to theta, and if you were to have your alpha power sort of shift over a little bit, it'll bleed into the theta range, causing the theta to sort of increase in power. So it's possible that what we see with the theta beta ratio is actually a sloped alpha. This is kind of getting to the nitty-gritty of like analyzing oscillations more carefully. But some more thoughts on this could be that there's a shift in the aperiodic spectrum. And if we sort of go back to this PSD, we can think about the aperiodic spectrum as to outline this portion of this signal that is sort of under this big triangle portion that is kind of like in most EEG signals. And a shift in this slope here could also give you a different integral value if you were to integrate over just the alpha range, for example. So you can imagine how like shifting this slope here can give you a different value as well. And those slope effects have been found to be correlated with a lot of things like aging. So there's still a lot of research on like what all these data analysis methods actually allow us to interpret about the brain. So do be careful, and also keep an open mind that these are not like fixed features that we can interpret from. And in the notes of these, I have like all the literature references and stuff, so you can like take a look at those papers, and we can send out the slides later.
Jiling, before you move on, we got a question that just popped up. It's asking, is the integral value is the integral of one particular range, right?
Yeah. So for the power bands here, it would be the integral for that specific range. But then because we're using power ratios, we're dividing by the integral over the entire thing. So when you say 0.5 to 30, that's the entire range. And then to get this like delta value, you would take the integral for just the delta and then divide by the entire integral over 0.5 to 30. Does that make sense? Cool, I think it makes sense. Feel free to ask more questions if you need. Yeah, so band ratios are interesting because of that. There's a lot of research into it. There's also, memory impairment has been correlated with higher theta gamma ratio, and you can probably find like literature on like a lot of correlations and things like that. But it's important to like look more carefully. There's not too much literature for motor imagery specifically for this, but it still might be worth exploring. So maybe it's like a pretty easy metric to sort of pick out from the analysis and code that we already have. So I encourage y'all to take a look at that. And if you don't find anything, that's still worth reporting. So yeah. Okay, so moving on from band ratios, we have another idea where we can take the channel differences. And this can be interesting because, as we saw before, the difference between C3 and C4 in, say, a power range can be interesting to look at. Because in the left-hand case, you get a positive subtraction between C3 and C4, but then in the right-hand case, you'll get a negative subtraction. And so that kind of emphasizes the difference between the two. And you can sort of think about how other ways you can find relationships between the different channels. That's just one example for that. Feel free to be creative. Okay. And then the next thing we have is time series statistics. These are a lot of things that you're already familiar with. You can apply standard deviation, mean, median, etc. on your EEG signal, on your PSDs, on your on the future things that we'll talk about as well with the instantaneous measures. So don't forget about all these statistics. Be creative. Think about how you can extract information about the EEG signals with these metrics as well. Then specifically for EEG, there's also a few other things that we like to look at. For example, first and second order differences. These are calculated by taking the next sample and subtracting that from taking the next sample and then minusing the current sample from it. So if you were to look at the EEG, the raw EEG over time, you can see how the first difference can represent how much it varies, like what the range of varying between time points are. And then same with second order, you sort of just get a slightly larger window of the time like differences. You get a very sense of the variability between the samples. And then there's also the Hjorth parameters, which give us activity, mobility, and complexity. We'll go over that a little bit more. And then also there's a paper here that kind of illustrates a lot of this math and how they applied it using machine learning to also do motor imagery classification. So if you have some time, you can also take a look at that. So the Hjorth parameters, activity is actually just the variance pretty much. So it's not super interesting. We already know how to get that. Two other parameters are mobility and complexity. Mobility represents the standard deviation of the power spectrum, and this is kind of like how it's calculated. It's kind of complicated, but basically you kind of want to see how much the power spectrum deviates over time, or not over time, but how much the power spectrum like changes in that one sample that you calculate the power spectrum of. And then this complexity is how similar the signal is to a pure sine wave. So kind of like if it's very similar to a pure sine wave, then you're just like, oh, it's probably not too complex. But then if it's more complicated, like it doesn't look like a sine wave, then we'll say that's more complex. So these are two other metrics that you can use, and we can, yeah, so y apostrophe t, that is the first derivative of yt. So y in this case would be your signal. And the good thing about using the PyEG library is that you don't actually have to calculate it. You just give it the signal and then they'll calculate the values for you. But yeah, there's a lot of good math behind this, and you can see them all in a lot of these papers. They actually like go pretty well and explain the different parameters pretty well with the math. So we encourage you to do that too. Cool. Good questions. Yeah, so that's the end of the time series stats and channel differences, band ratios, and power band analysis that we can do with PyEG and like traditional NumPy and things like that. Are there any questions about these so far? Oh, where can we find the slides? Simon or Darren, would you guys, one of you guys be able to grab the link, like find a view-only link? For me, I kind of, I went to a bunch of lab meetings. The Wojtek lab meetings were super helpful for learning these. I also took signal processing as as a younger undergrad, I took EC 45 and 101. Those are pretty helpful. But these are like an application of those informations. And I guess it's not 100% necessary to like know all the signal processing, but if you're interested in learning that, then yeah, those classes are really helpful for getting a better grasp. Most of these are kind of just methods that you can see in literature. So if you were to like look up feature extraction for motor imagery, for example, then you can find like power bin analysis, Hjorth parameters, first order differences, and also sort of just exploring PyEEG. You can find a huge list of other features that you could potentially use, and then trying to map that to literature. And then there was one more thing I wanted to say. Yeah, and then kind of creativity is another thing, is sort of like understanding how this neuroscience works for this, sort of going to COGS classes and learning about electrophysiology in a lot of the cognitive science literature. You can sort of use your creative understanding of statistics and signal processing to come up with new ways of featureizing things like this. So in the sense, it's actually a very creative process too. Cool. Yeah, so hopefully that wasn't too much information yet, because we still have some more. So we'll also take a look at instantaneous features with NeuroDSP, which is the digital signal processing toolbox that actually here at UCSD, the Wojtek lab developed. So these are just some of the things that you can do with it. There's some more, but we won't go through all of them. In particular, we'll look at the phase and coherence of neural signals. And to get this, we have 3 metrics or 3 time series values that we can extract from our EEG data: amplitude, phase, and frequency. And in literature, EEG data has a lot of like phase amplitude coupling things associated with it. And just to give you a visual as to like what that means, if you have phases that couple together, then you kind of think about that as being kind of like this, where you have sine waves that are at the same phase at the same time. If you have phase amplitude coupling, you might think about how having a large phase value can also give you a large amplitude value during certain times. So being able to identify these type of patterns is also very interesting. And there's also like amplitude correlation, where you might have like different frequency bands that have amplitudes that correlate. So that might be interesting as well. Similar to that, there's a phase locking value that is explained in one of the grad students, or graduated now doctored students at the Wojtek lab, and I have linked the video on actually a lot of these things in the notes for this slide as well. So if you're interested in his explanation of that, it's a really good, it's like 30 minutes long. And then there's also instantaneous metrics for motor imagery in literature. So we can see some people have tried to use instantaneous frequency, amplitude, and phase to better their models as well. And the literature is also in the notes section. The analytical signal is a breakdown of the signal to amplitude and phase. And this is kind of done by getting the complex version of the signal by using the Hilbert transform. And then the analytical signal is basically like, you know how the complex domain is like perpendicular to the real domain? You have an amplitude, which is the length of the ray that sort of comes out from the 2 real and complex components. And then you have phase, which is the angle from 0 that the signal is currently at. And so these are the values that we're basically extracting for our signal in the instantaneous domain. And that was a lot. Yeah, we'll get a visual of this, so hopefully that will make it clear. So amp by time, you can sort of think about it as first filtering the band to whatever band you want. So in this case, alpha power, we filter it into just the alpha power, so you can see kind of a more smoother signal down here. And then what the amp by time does is that it basically hugs the signal on its amplitude over time. And so you can sort of think about how like this can be helpful. It's kind of like taking a slice of the spectrogram in the I guess time domain, where if you were to take it over the alpha band range and then flatten it somehow, then you would get something like this, where you have a representation of how much power there is in alpha over time. So this one should be like kind of like the easier ones to understand, because you have the actual signal and then you can visualize the filtered signal, and then you can visualize that the amplitude of that is just the thing that hugs the outside of the signal. Cool. And there's also phase by time. So this is calculated similar to the amp by time, but instead of taking the amplitude, you take the phase. And here on the right, you can see a visual of what phase means. If you have a sine wave, then you have 0 be at, like, if this is cosine, if you have a cosine wave, I guess, then when cosine is at 1, you get a phase of 0, because at the 0 degree line, if you think about the unit circle, you get cosine is equal to 1. And then when cosine is equal to, say, or -1, then you get the value like kind of swap from pi to -pi, or yeah, it's a little complicated in this like long part, but you can sort of just imagine the angle sort of increasing over time, but then like goes back to 0, right? It has to like somehow like swap back. So you can think about, yeah, if you're at cosine equal to 0, you get some value here, and then you keep going, and then it just tracks like what the phase angle is as this unit, as this array sort of goes around in a circle. So this is kind of like what it's doing here. If you take the filtered signal again, you can sort of get a measurement of what angle the signal is at currently. So you can see how there's like some blips sometimes. Sometimes these values get stretched, or they get squished, things like that. So if you have like this kind of metric, you can sort of couple it with some other metric and then see how that might give you another feature. And then there's also frequency by time, which is actually defined as the derivative of the instantaneous phase over time. This one has been used to identify sleep stages and also seizure detection. So yeah, it has like pretty good literature on several mental states. And also there is some literature on motor imagery using this metric as well. So that's pretty interesting. If we look at it, we can see how the phase over time on the bottom, when it sort of, this like normal-ish value, this like flat line, is kind of like what it's supposed to normally be. And then whenever it deviates from that normal-ish phase that we see for a specific like say frequency, then we'll see these like little blips come out of it. The definition of phase, it's sort of defined by like the angle by which the vector that creates your signal, what angle that is to the 0th line in the unit circle. So if you think about like the cosine at whenever the ray is pointing directly to the x direction, where it's 1, 0, then you get 0 angle. Yeah, so if cosine is 1, cosine of x is 1, cosine of 0, where x is cosine of 1, which is the x value of wherever this ray is, that's equal to 0 because the angle is 0. And then if you like continue doing that, you get the phase at like whatever location the x value is for cosine. So if you can sort of see here, I guess, you're just imagining how a sine wave works and how like over time you have, it goes from like negative pi to pi or 0 to 2 pi over time, then it just kind of oscillates in these radians. Yeah, it's like the angle of how far it is from the like line that creates the x-axis. Yeah, so calculating these, you can use very simply as statistical, like you can put statistical things on them, like standard deviation, means. Those are the easier ones to do. But if you want to do like phase amplitude coupling or phase locking value, those have like some more equations and stuff. So you might need to look them up or find some package that does that. Yeah, or you can like come up with some creative way to like think about how, like, say the amplitude is increasing at a certain point in time, if the phase is also increasing at that time, or in this case, if we thought about the phase amplitude coupling, then say if the phase of a lower amplitude or phase of a lower frequency band is coupled with the amplitude of the signal at a higher frequency band, then you might get something that looks like this. So you can do this across the frequency bands, potentially across channels too, actually. But that might be going a little bit far out there. Cool. Yeah, good questions. Okay, so yeah, that's the end for the like talky part of slides. If anyone else has questions, I can continue taking those. But for now, we'll take a little bit of a break and let people set up for the interactive track. The only thing you'll need to pip install new is the PyEG thing. And that one you actually have to do this like special pip install because they don't have the PyPy set up for it. So make sure you do that one instead. And then, yeah, if you haven't gotten the notebooks yet, feel free to git fetch, git clone, and then we'll get back in like a couple minutes. So 40, I want to say 45 because that's a nice number. But maybe 45 or 46 or 47 or something. So yeah, feel free to take a break for 4 minutes. Cool. So we got a question from Anish about whether the machine learning model we'll be training on is going to be on raw EEG data or a mix of, or just the features that are being extracted, or a mix of both. So that's a good question. I think traditionally with more traditional machine learning models like LDA, SVMs, those that sort of like split lines between feature spaces, it's a lot easier to give them just the features that we're trying to extract now, because these features sort of represent the data in ways that are more handleable than giving it 1,000 time points. Because if you give it 1,000 time points, that whole signal could basically be identical to that whole signal, but shifted over by like 1 time point. But then you would have like completely different actual like voltages and values for those 1,000 time points. So usually for these types of traditional models like LDA, SVMs, we will only want to use the features that we're being extracted. And just to say a little bit more about that, we also don't want to give it too many features, because if you don't have a lot of trials, having too many features would actually easily overfit your model, because just thinking about how, like if you have, say, just 100 trials, for example, you guys have a little bit more than that. But if you also had 100 features, then basically each feature, like you can model exactly like how these trials can fit into classes with these 100 features or so. You can always find something, or it's much more likely that you'll find something that does not like extrapolate for the rest of the data set. So that's another reason you might want to only find the features that actually differentiate these classes better or best to fit to these traditional models. And we'll see more next time as to how you can do that and how you can like try to identify the ones that are like more differentiating. For just fitting in the raw EEG data, those do exist if you have like neural networks and things like that. So if you kind of, I think the deep learning workshop from DS3 could potentially kind of go over that. But usually CNNs and things that have like ability to take in like all the pixels of an image would be able to do something with the actual just raw EEG data. So those two methods are used, depends on your model. Cool. I think we are pretty much, okay, yeah, we're right on time. Yeah, if you use neural networks and like EEGNet or Brain Decode, things like that, those do just take in raw data. And when I say raw, you do still have to filter it, because EEG data, we didn't really go over this because this entire data set is already filtered for us. But if you do record your EEG data from scratch, from raw, you'll get a bunch of 60 hertz noise. And if you throw that into your model, it's not going to do anything. So yeah, you also have to filter it, but after you filter your raw data, you'll be able to throw it into something like that. Yeah, and there has to be like specific ones that work for EEG data, because we are primarily interested in like spatial and temporal differences. And so the convolution in these two dimensions is very important. Cool. Well, all right, so we'll just do a notebook walkthrough. I guess, can we get a show of hands as to how many people are following along with the notebook? Cool, 3 people. Awesome. It's great. Yeah, if you don't have a setup yet, don't worry, we'll have a setup thingy, and yeah. Okay, you can still see my screen, right? Okay, awesome. So if you have the notebook, it should look something like this. If you have the data in the folder, you should be able to run it. And we can see that over here we have our imports. We have our constants, which tell us which channels are what. We have a single helper function. I got rid of the ones from last time because we didn't really need them for this workbook, but it could be helpful to use them as well for your analysis. We load the epoch data just to keep things simple. So just as a refresher, we have each row be a trial. And this is the ID of the patient. This is the start time of the trial. This is the event type. So 0 is left. And this is the data. And each of these arrays is like 1,000 time points. And there's some EOG data here, but we'll kind of ignore that for now. There is a bonus workbook that I'm working on for extracting EOG from, so basically iArtifact information from the neural signals, but that's not necessary, so we'll just ignore that for now. PyEG features, these are just some helper methods for getting the power ratios and things like that, and intervals. Okay, so when we want to get the power bin, we kind of want to do this for every single trial. So you have a signal for each trial for each channel, and we get the power ratio of that EEG signal for that specific binning, and this is the binning I chose because this is traditionally like these bands are used, but you can also experiment with like smaller bins, larger bins, maybe go higher or lower, maybe only look at certain ranges more closely. But yeah, it's like up to you how you want to change that. And then you want to go through each channel because you have kind of a signal for each channel, right? And you get the ratios for that. And then for each of the intervals, you basically try to assign this to this power ratios dictionary that will keep all the information about the channel and the frequency band and the value of the power ratio there. And after we do that, we get this data frame that has all these values. Yay! Does anyone have any questions on how, like any of these parts of this thing? Because this is kind of like pretty fundamental to how you might like extract features in the future as well, because you can extract other features. Right here is, this is like this ratios thing is like the specific part of this that is actually different. Oh yeah, get intervals. Yeah, so this one actually is kind of like just a nice one that allows us to create these tuples from the binning thing. So this binning thing is not super helpful in like trying to create these like just tuples. So get intervals, actually that's the only purpose of it, is to create these tuples so we can label things. Yeah. Yeah, it's yeah, it's good you're looking in details. Cool, yeah, if anything else comes up, just feel free to put it in the chat. We do see something very similar here when we want to, oh, actually, wait, yeah, we're still working with power ratios. Okay, so continue with power ratios. Not only do we want to get the averages, but we also want to get the standard errors. So here we're going to basically analyze this between classes again. We want to see these graphs, basically the ones that I showed earlier. And so when we take the averages, we basically just take the average of all of the y's that are equal to that y. And yeah, that'll give us the average. And then, however, we still want the standard error mean because we don't want to like just assume the average and say that, oh, there's a difference in the average, which means that it's some significant difference, because it's possible that you have such a large fluctuation in your data that it actually doesn't mean anything to have like that tiny bit of mean difference. So you want to have the standard error mean also be calculated and plotted here. So you can sort of see that, oh, okay, there's like a tiny standard error mean, and that's smaller than the difference. So we can potentially use this as a factor for differentiating two classes. Like over here, we might see that's probably not as helpful. And you might want to graph these bigger because they kind of graph them small. But yeah, this will do that for you. It'll calculate these standard error errors and averages and create the standard error data frame and the average data frame. And then we could just plot them with this plot multiple bar graphs. It's kind of complicated to use. I'm sorry, I wrote this function, and yeah, it's not the best function that I could have written, but this is kind of how you use it. You put in the some information of what you want to plot, like what the actual values are. And then you have, this is like the bin width, and then you have some labels for like the different classes. And then you have the intervals, which is these labels down here. And then you have the error values, which is the other values that we calculated. So standard error goes here, averages go here, and then you get to plot these. It's probably clearer if you just look at the actual helper function. But yeah, feel free to like rewrite that or do whatever you want with it. Cool. Yeah, so that's how you create these graphs. Are there any questions on that? Cool. Okay, let's continue. Power band ratios. This is very similar to the power bins that we calculated, because we kind of go through like each channel again. Well, this one, it goes through all of the power ratios that we calculated and tries to find the ratio between the theta and beta power for a specific, or for each of the channels. So you can see how you have to construct this dictionary. This dictionary gets created, you get something like this. And then if you want to concatenate it to your original data frame to create the feature data frame, then you'll have this. Woohoo, more features. And then channel relationships, also very similar, except you're doing differences. And you see we iterate through different things to get this. I'll let you kind of dig through that code, I guess, because it's pretty similar. But now we have like all the differences and everything too. And then for the first and second differences and Hjorth parameters, I kind of let them be for exercises for all of you, because it's pretty much just a different call function to PyEG. And there's also other stats you can think about, analyzing temporal and frequency and spatial information. So highly encourage y'all to experiment with more new things. Down here, okay, yeah. Are there any questions about that so far? Yes, there will be a new notebook for the next meeting. Yeah, so this code, it's kind of like a mix. Actually, a lot of this is just like vanilla Python, like all the looping and stuff. This function that I wrote up here for the plotting, it's a mishmash between Stack Overflow and stuff that I've used in the past for my projects. So it's kind of readable, at least for me. I don't know about for you guys, but it's not the most like easy to pick apart if it's your first time plotting with Matplotlib. Loop more efficiently. Are you talking about the here with the calculating the sems and the averages and stuff? Yeah. Let's see. So if you think about calculating Sems and averages. If we were, I don't think you could do this in the same loop here, because first you need to get all the values, and then you can take the average, unless there is a way to like take an average while you're, I guess you could sum everything up while you're doing this. So if you're going through this and you know the event class and you know like how many elements there are in that class, you can keep running sum and then just divide that to get the average later. So that's possible. Yeah, you could do the same. Wait, actually, can you do this? No. Well, you'll need a list. You'll need a list for the sems. So you'll still need to finish this and then calculate the standard error means. But this over here is actually just looping through the different columns, basically. So it's not looping through like all the trials again. And then over here, if you want to plot them, I guess it's easier to plot if you have all the values. So yeah. Yeah, I haven't gone through that yet. Michael, you're going really fast. Yeah, we'll get into that right now. Okay, so NeuroDSP. This is a subset of the library. So if you want to explore more, the paper is here, and I think I linked the link to their GitHub here. PyGs is here, and then some more papers that are good, and then experiment setup. So you can feel free to explore that. But if we want to go through the amplitude by time, let's keep the sample and let's only look at a single signal. So we're only going to look at the first trial and at C3 electrode. And we also have alpha range, which is defined as 7 to 12 hertz. And if we want to get the amplitude by time, it's actually pretty simple. If you import amp by time, all you do is pass in sig and EEG-FS and the alpha range that we just created, and then that will give us the amplitude by time. And you can plot that. One caveat is that you'll actually get some nans in the beginning, so it's not gonna match exactly. I think it has to do something with the Hilbert transform at like the frequency domain, because you need some signal in order to actually calculate things. You need like a window of signal, and so like some of these values just won't have anything. You could like zero pad it or something to make your signal longer, and then match the non-nan values to get to the same length. But I didn't do that here, so that's up to you if you want to explore that. There's like different ways you can pad, and you'll get different values too at the ends. So yeah. There's also phase by time. This is very similar. You just use the phase by time one. Yeah. Actually, these are like not too interesting in terms of implementation. So yeah, these are just nice to play around, to try different signals, try different ranges, and get a feel for like what these are, and then see what you can extract from them. If you were to loop through, calculate all these things for all your different trials, and maybe then calculate some other feature, like how do they couple together? How do they, what are the statistics, like standard deviation, things like that. And yeah, so that's pretty much the whole notebook. There's a lot more for you to explore here than last time, I guess. But yeah, I think getting a good idea of how this code works and how you can implement future stuff would be really helpful. Are there any questions about the notebook?
So when you say calculate the mean or standard deviation, are you talking about the mean across trials? So you get like, dampened wave, or are you talking about the mean across the time across all trials? So you get a single number? What does that exactly mean?
Right. So when we think about standard deviation of any set of samples or time series thing, well, when you have a time series, you basically have a bunch of samples, right? Standard deviation is kind of how much it deviates from the mean and like how wide that thing is. So in this case, if we were to apply this to the instantaneous frequency, we can see that this signal might have some pretty large standard deviation because we have some values that are fairly far off from the mean. And comparing it with other trials that might have a smaller standard deviation for this frequency by time series sampling. And then you can do the same thing for this, and then amplitude as well.
But again, this is just for a single trial. So would we just kind of concatenate all trials together and then get the standard deviation across all?
Oh, yeah, to clarify that, I would say that you would be trying to aim for features for a single trial. Because you want as many features for a trial as possible. Like you want information about your EEG data for that one trial in order to be able to classify it later on. You could find the standard deviation if you were doing more of a, I guess, cognitive neuroscience analysis, if you're more interested in the neuroscience aspect, which is also a very valid approach to this competition, is to, yeah, as what you said, to do the statistical analysis on like the entire population of trials, because from there, then you can make even more some conclusions about like the patterns between the two different classes.
Okay, I see what you're saying. Okay, thanks.
Yeah. Cool. What's the intuition that we get from amp by time? Yeah, so that's this one over here. As we sort of saw before, I think this one might give you better intuition. It's kind of like when we saw the spectrogram last time, we have something over time, like how much power there is in every single frequency band over time. In this case, we want basically how much power there is in a specific frequency band over time. And this kind of gives you a little bit of a better handle as to how you can featureize the data, because instead of having entire other dimension of spectra of frequencies, then you can only focus on this one like smaller range of frequencies and then get how much power there is, or how it's pretty much directly related to how large the amplitude is for the EEG signal at that time. So if you compare it with like the original EEG signal, you can see how it's actually pretty close to how big the up and downs are for this part. And if you like imagine taking a lower frequency band versus a really high frequency band, you can get different amplitudes over time. Hopefully that made sense. And phase by time. Okay, phase by time. Yeah, so I think the best way to understand the phase by time might be to sort of think about this diagram again with the neuroscience. If the two signals, if like say two power bin signals are correlated, or maybe two electrodes at a certain time are correlated, for example, then you might think that those two circuits in the two regions are kind of firing together. It's like a pretty broad generalization, especially with EEG, but like if you were to go back to the neuroscience, kind of what it would mean is that the recordings of the two neurons are kind of in sync versus not in sync. And so if you have phase by time information, then you can compare that with, say, another channel's phase by time and see how in sync they are. And then that can give you information about how correlated those two brain regions are. Cool. Thanks for questions. Okay, so I think we'll wrap up the notebook walkthrough with that. Just to remind everyone about upcoming events. We are having a de-stressor social next week. It's going to be super cool because one of our officers actually does a lot of meditation and he'll be teaching you about how to like sort of de-stress in this very stressful situation right now. And so even if you're stressed about midterms or anything that's happening right now, feel free to come chill with us. We'll also have like a breakout room for people who want to actually like socialize with people interested in neurotech. So if meditation is too boring for you, or you've done it before and doesn't work for you, then feel free to still come and we'll try to entertain you with games or just chatting. So it's just a very casual, just come by and then get to know your fellow neurotech enthusiasts. And then we have EEG feature extraction 2 workshop, and then another event coming up, and then we'll have some more events and the competition work wrap up in the winter quarter. Okay, so I think that's it for the actual workshop. Thanks for sticking around a little bit extra. If you guys need any of these links, these are here. But yeah, we have question office hours now. Yeah, so if you have any questions about the competition, feel free to go to the NeuroDataset competition or the Neurotech at UCSD Discord, and then find me or any of the officers, and then just like DM us for specific questions. Or if you think that the question might help others, you can also put it in the general chat or the NTX neural data competition chat. We could also create a questions channel. That might be a good idea. Yeah, if you DM me the question and I feel like it's a really good question, then I might have you also post it in the questions channel. That might be another way to go. Cool, yeah. Thanks everyone for coming. Yeah, if you have any more questions, we'll just be here. Yeah, no problem. Oh, I guess if anyone doesn't have the sign-in link, it's over here.
Geez, G-Link, easy on your keyboards.
It's a new laptop. I can take it.
I know it's the new laptop. Take it easy.
It doesn't have butterfly switches.